thank you all so much for coming today. I'm really excited to share some present research, my lab, our research, and our interests, and our future directions. Also, this should probably say, what can cavefish tell us about carbonate, karst, aquifer, connectivity, but I tried to keep it a little bit shorter than that. It's on the iPad. OK. So as mentioned, I am a new professor in biological sciences, and our lab is the Extremophile Evolution Lab. I have shortened it to Extremo Evo, but Twitter dubbed us eel. And since we study fish, they were like, oh, you're the eel. You're the eel lab now. So we'll see which of that sticks. But we mainly focus on the evolution of fishes in the dark, and this includes cave fishes as well as anglerfishes. Today, we will be focusing on the cave fishes. My professional scientific career actually started right here in Alabama. About 10 or 11 years ago, I was at that other SEC school that's in Alabama that I'm not allowed to say. My undergraduate advisor brought me up to caves in Huntsville. And this picture was actually my first caving trip. That is a Ziploc baggie with cave fishes in it. And this was the exact moment that I decided I wanted to do this forever. Experiencing these caves really firsthand and seeing the profound variety of organisms that live and thrive in these environments has really forever fascinated me, and this has driven my research. When I'm talking about extreme environments, we talk about places like deep, fast-flowing river channels, deep oceans, caves, and these are places that are geographically isolating, and they impose harsh physiological demands on the resident organisms. Extremophiles have complex innovations that allow them to live in such harsh environments. We have antifreeze glycoproteins in Antarctic fishes, cell walls or sheaths in thermophilic bacteria, eye and pigment changes and bioluminescence in the deep sea, as well as eye and pigment changes in caves. Some of these extreme environments have the additional pressure of darkness, and that is where most of our work so far has been. Innovations, convergent evolution, harsh habitats, these have really inspired the research for my lab, which is meant to understand organismal evolution to these environments. To address this, my lab has three main components. Firstly, to document and conserve biodiversity. Secondly, to clarify relationships across tree of life. And thirdly, to investigate some of these innovations of extremophiles. As mentioned, our lab primarily studies cave organisms. Caves are great systems to study adaptations to extreme environments. They are dark, cold, and there are scarce resources. Organisms adapted to life completely in the cave are characterized by the loss of eyes and pigment, as well as enhanced sensory capabilities. And I was actually trained traditionally as an ichthyologist, so fish are pretty much our focus. You may be surprised to learn that there are around 170 different species of cave-adapted fishes, and they span across the fish tree of life, including 10 fish orders. In particular, my lab studies the North American Amblyopsidae. This family includes closely related surface-dwelling species that have eyes and visual perception. And then we have the cave-dwelling species that lack external eye characters and the visual perception. This gives us all levels of cave adaptive evolution, which provides us a beautiful natural lab for understanding cave invasion, but also, as they are fish, for groundwater and aquifer invasion. The family is distributed throughout the southeastern United States, along the coast into the Ozarks. And there are cases of co-occurrence where these fish are found together. And not just between surface fish found outside the cave and cave fishes, but rather there are areas where you can find a single cave with multiple species of blind cave fishes from this family. This distribution map also closely mimics another map that might be familiar. This is the Karst map of the United States, particularly pointing out carbonate karst aquifers. And you may see that some of the distribution in the Ozarks, in the southeast, on the coast, corresponds to where these fishes are. Karst landscapes are characterized by dissolution of host rock and can be carbonate, which is limestone, and cavey. We call it cavey. I am fortunate to go into the field, and finding these fishes can be difficult, not just physically, but the fact that the fish aren't always there. Sometimes we'll go into these caves. They're historical localities. So someone at some point in time has found a cavefish there. We will go in, maybe the day before we found a cavefish there, next day, can't find any. What we think is that these fish are actually living most of the time in the aquifers that can connect to the caves, the groundwater connected to these caves. And we will revisit aquifers a little bit later. Through collaboration with caving groups, recreational cavers, landowners, and state natural conservation resource managers, we have been able to set up this study system as a model system. So outside of capturing difficulty, the family really seemed like a great system to study fish distribution, fishes going underground, all of these aspects. But they unfortunately have a complicated evolutionary history. These are estimated evolutionary histories of species represented in a tree-like form, and we call them phylogenies. We read them from tip to root, and I kind of explain phylogenies a little bit like a pedigree or a family tree, but just zoomed way out. So these are not individuals that we're connecting via, ancestors or grandparents, but rather they can be species, and we are looking at deep evolutionary time. So, based on the data, including how these fish look, but also their genetics, and based on who is analyzing the data, we get a bunch of different evolutionary histories and relationships. And this muddies the water as to how cave fishes actually got underground. Whether it happened once, twice, multiple times, we're not quite sure. So in this project, we were concerned with eye trait loss, but also that corresponds for us to groundwater invasion. So our questions here: how did these cavefish lose eyes in conjunction with invading the groundwater? Understanding how these fishes got underground may actually help us understand the entire system and groundwater that they live in. We have 3 hypotheses based on these for how these fishes got underground. These are our 3 hypotheses, and I have little infographics as well as example evolutionary relationships to kind of step us through how these fish lost their eyes and got underground. I apologize, I am not a graphic artist, I am a biologist. I did my best. So our first hypothesis is known as progressive regressionism for eye loss. This is a slow progression from an eyed ancestor that then becomes smaller eyed, and then finally we have a single subterranean invasion followed by eye loss. So in addition, with a single cave invasion event, we then must have subterranean dispersal. So these fish have to somehow, when they get underground, then be able to speciate and diversify, because we have multiple species of cave fishes. What this looks like with our tree, our phylogenetic tree, what's really important here is the eyed fishes in relation to the blind fishes. So our eyed fishes are colored in orange and our blind fishes in blue. We begin with our swamp fishes. These are surface fishes, and I love that they're named swamp fishes because they live in swamps. Finally, a scientific, somebody is making sense here. But then we get confusing. Then we have eyed spring cave fishes. These cave fishes do have eyes, as I mentioned, and they live part of their lives outside of the caves and part of their lives inside of the caves. Next, we have the cave obligate or eyeless fishes. Because the cave fishes all share a common ancestor in this particular tree, we have a single subterranean invasion by an ancestor, and then this leads to subterranean diversification. Our second hypothesis is eye redevelopment. Now, I know this sounds ridiculous. I'll explain. This hypothesis does suggest that a surface fish invaded the subterranean habitat, lost eyes, and then recolonized the surface habitat, regaining eyes. There has actually been evidence for complex trait loss reversal in organisms, including snake oviparity, so giving birth to live or eggs, as well as salamander metamorphosis, so going from juveniles to adults and keeping some of those characteristics. So there is some evidence for complex trait loss reversal. This was supported by previous research with what's known as ancestral state reconstruction. So trying to identify what the ancestor, if they had eyes or not. So they found support for a blind ancestor here and eye redevelopment or surface recolonization with these eyed cave fishes. So they found the cave fishes nested, the spring cave fishes nested within the group of larger blind cave fishes. Lastly, we have multiple eye loss events where we have multiple subterranean invasion events, multiple times going into our groundwater, and multiple eye loss occurrences. Some gene mutation data related to eyes, as well as looking into dissection of eye characters, suggest this multiple independent cave invasion rather than eye redevelopment. So we still have the eyed spring cave fishes within the group of larger blind cave fishes, but we still have an eyed relative and multiple subterranean invasions. The overarching goal of this project was to examine evolutionary history and trait analyses in extremophiles in groundwater, particularly using the amblyopsid cave fishes as our study system. Our two main objectives was first to clarify these evolutionary relationships, understand how these fish are related, then to determine the most likely scenario of groundwater and cave invasion. So these are our evolutionary relationships. I can get into the data set if you want later, but this gives us our interesting relationships in which we will go zoom in. We recovered the eyed spring cave fishes within the group of larger cave fishes. So then this could either mean we support surface recolonization or multiple independent eye loss events. So we tested multiple models of evolution and found that we found the most support for the irreversibility of eye loss. So that means that we did not find any evidence of a regaining of eyes. So this means that we have multiple cave and groundwater colonization events at these 3 arrows. So for this particular project, we found the eyed spring cavefishes were nested within the blind cavefishes. We resolved competing hypotheses of colonization into the groundwater habitat, and this in turn means that the cavefishes did not solely diversify underground in the groundwater, but rather each 3 ways we have the consequence of multiple invasions. So we just talked about them getting into the groundwater in the first place. But once they're there, what then happens? We can go from the whole family to one genus in particular, genus Typhlichthys, and isn't she a beauty? So this is the southern cavefish, and currently we say that there are 2 species within it. However, we're confusing. We, as biologists, we love to categorize things, and when we can't, we get very upset about it. So what happened was, in the early 1900s, they were described as separate species. Then in the 50s, somebody was like, no, they're not. So they're sunk back in. Then in 2013, the name for Eigenmann's cavefish was literally resurrected. These fish were not redescribed, though. What that means is that we didn't have any morphological or physical characters to define them as species, but what we did have was DNA that told us they were on their own evolutionary trajectories. However, since we didn't have any physical characters to call them species, they are still considered part of what's known as a species complex. What's really interesting here is the distribution. If this is all the same genus that we expect, how did these fish end up like this? These are the cave obligate organisms. They are distributed across multiple states, and this means across multiple drainage basins on the surface, but this also means across possibly aquifers. So we thought that this would provide a great opportunity to examine aquifer connectivity on a wide scale. So putting the pieces together via how the fish are related and how, if they travel. We thought it was a good idea. So how did this complex get to be this way? We have 2 hypotheses. You may recognize these hypotheses. Either we have a single colonization event into the groundwater, followed by underground expansion. We know what this looks like. Or we have a potential widespread surface ancestor that colonized the groundwater multiple times. Then the surface ancestor went extinct. These fish do not have a counterpart on the surface. They don't have a sister on the surface. So it may surprise you to learn, but cave connectivity and some aquifer connectivity is widely unknown in the southeastern United States in relation to caves to aquifers. Since we don't have a great grasp on the cave connectivity, and the only way we can find these fish is via the caves, we have to kind of think about it in a cavey way. When you think about caves, you think about them as Cave C here, very insular. They're their own particular habitat, not connected. That's what we usually think. However, there may be some ways that caves are connected. There are fissures in rock that can create a system where dispersal and then possible interbreeding can occur. So the extent to which cave organisms can actually make it through some of these networks is hotly debated in cave biology. We also, because we don't quite understand cave connectivity, we don't actually know what a population of these cave fish is. A population is defined as individuals in the same time and place such that interbreeding can occur. So place is important for this. That means we have to kind of know where the boundaries are. And not knowing how these caves are connected, we don't know where the boundaries are for these fishes. This, I just loved, was a wonderful schematic of visualizing the systems a little bit better. We have evidence that these fish do primarily live in the aquifers that can be connected to caves. They are found in people pulling up well water. So dug wells, we imagine, and in some cases there are no caves nearby, but you can still find cave fish. So we have the increase and decrease of the water table, and it's possible that some of these connections between caves can become filled. Could fish go through it? Maybe. Or are the cave fishes all connected via the aquifers? Those were some of our questions. What is a population in these cave fishes? How are caves or caves and aquifers connected? Also, how are the populations related? By examining the history of these cave fishes via whether they disperse via the subterranean habitat or not, can we possibly understand a little bit more about aquifer connectivity on a wide scale? This is the distribution of the complex again with our sampling localities, showing that we did cover the range of Typhlichthys using both the southern cavefish, which is in the southeastern United States, and Eigenmann's cavefish, which is in the Ozarks. This was a massive sampling effort over 13 years in which I was grandfathered in some of these samples. So this is a massive collaboration with cavers, caving groups, landowners, and state resources. Okay, this is a genetic clustering method, and this both includes the southern cavefish and Eigenmann's cavefish. They are not labeled yet. This is created with a data set of common population genetics data called single nucleotide polymorphisms. And I may switch between the terms genetic cluster and population here. This particular method, because we have to argue it about everything, population geneticists argue whether these blocks are considered true populations or if they are just how the genetics cluster. So I may switch in between them. I'm going to use them synonymously today. Each bar is one sample, so one individual. And the amount of color indicates how much of the genetics was assigned to that cluster. So if we have a full bar, that means that 100% of the genetics is assigned to that cluster. Our most likely number of genetic clusters was 5. And what I want you to notice here are the extremely well-defined blocks. We do not have any individuals that have 50% of one color and 50% of another. This means that there is not a lot of genetic exchange between these clusters. So when I saw these data, I was expecting we have the Eigenmann's cavefish is going to be one of these clusters, and then the rest are going to be the southern cavefish. They are supposedly, distinct genetic lineages, separate. That is not what we found. Instead, we found Eigenmann's cavefish clustered with individuals of the southern cavefish. So nested within the southern cavefish. And that means that there are Eigenmann's cavefish that are more closely related to southern cavefish than southern cavefish are to each other. So within the same species versus between species. So next, thinking about geographic distance, gene flow, water connectivity, I thought, all right, maybe Eigenmann's cavefish that are clustering with the southern cavefish are geographically closer, and perhaps that allows for some of that gene flow. Maybe those are those aquifer connections. I did not find that. The geographically closest southern cavefish to Eigenmann's cavefish are not in the same genetic cluster. So by distance, the closest southern cavefish genetic cluster is purple versus the blue that Eigenmann's cavefish connects with. So these data had some surprises, and those surprises just kept coming. Here, I want to point out the localities. So each of these is a sampling locality. The localities that are pie charts instead of solid dots. Those are instances where we found specimens from the same cave that grouped with different distinct genetic clusters. So there are multiple caves in which this occurred. I've pointed them out in yellow, and then I have highlighted some of one cave in particular, Bobcat, just to show you the difference in genetics. So this finding supports that a cave is not a proxy for population, as we find multiple genetic groups in the same cave. So they're not sharing genes. What is the reason behind this genetic clustering? This was the crux of my question. More specifically, do these 5 clusters match up with geographic boundaries? Do they match up with surface drainage basins, surface drainage subbasins, or, for me, most importantly, carbonate aquifer limits? And I was sure. I found absolutely no relationship between genetic structure and these karst aquifers. So genetics does not align with aquifer boundaries. It also doesn't align with surface boundaries. Moving on from geography, we also looked at how these populations were genetically related to one another, how these clusters were related. I am really passionate about conservation. These are wonderful and rare fishes. So these were particularly for conservation estimates. What we found is that we have high genetic structuring, but low genetic divergence and diversity. These estimates are actually pretty concerning for conservation, as high genetic structure but low genetic diversity indicates really small effective population sizes. So this is a small group of adults that can actually reproduce. That is not great for the continuation of a species. So we were able to find some interesting conservation and biodiversity implications. Coming back to our objectives, we found evidence that one cave is not one population, as we found individuals from the same cave assigned to different genetic clusters. Low genetic estimates means less genetic material for adaptation. So it's more difficult for the organism to adapt to stressors, easier for them to die out. That's why these estimates are actually concerning. The result that caves are not a proxy for population also speaks to our second objective, in that there may be some level of unknown cave connectivity facilitating genetic exchange across these carbonate aquifers. However, since there's no genetic exchange between the clusters, so we have the clusters that spread across aquifers, possibly they can maybe interbreed, but between these clusters, we don't see any genetic exchange. What we think is that that supports multiple independent invasions by the widespread ancestor. To speak a little bit more to this, even though we did not find a structure related to surface basins, so it's not lining up with the surface basins. What is interesting to note is that the distribution of these fishes does follow that of some of the major rivers and streams in these areas. So we don't actually think that these fish are crossing the Mississippi. It's more likely that this wide distribution occurred via the surface ancestor. So to actually answer the question, what can cavefish tell us about karst aquifer and carbonate karst aquifer connectivity? They may not be as hard of genetic boundaries as personally I thought. And these fish may be able to move between these aquifers without as much trouble. While conversely, we also have multiple groundwater invasion events that possibly speak to the massive distribution of these fishes. I mentioned conservation with population genetics estimates, but we're also interested in broad-scale groundwater and cavefish conservation as well. And that includes anthropogenic stressors on groundwater. So these are some of our future directions. Groundwater supplies 37% of public supply water around the world, and karst aquifers specifically provide about 20 to 25% of drinking water around the world. And groundwater pollution and exploitation are major issues. In addition, we do not understand how these anthropogenic stressors will affect these systems. We particularly are interested, as mentioned previously, about innovations, and one of those innovations in these fishes are their enhanced sensory capabilities. So putting these concepts together, how are the sensory systems of these fishes impacted by environmental stressors from us? So just for a quick second, I'm going to talk about our future water quality, specifically water quality research. How does water quality affect sensory capabilities of cave fishes? We already know from surface fishes that heavy metals, noise pollution, increased turbidity can all affect sensory capabilities. It messes up their signals. This includes things like chemoreception, so that is taste and smell, and also what's known as mechanoreception, which is sensation of touch, sound, or changes in pressure. Fish feel these mechanoreception using what's known as the lateral line system. This is a system of hairs distributed across the head and body of these fishes that allow them to detect the movement of the water. They're also very similar to human inner ear hair cells. We know from the single model cavefish, which is a blind cave tetra, that they have more sensory units than surface counterparts. And we also know that they have more cells per unit than their surface counterparts. These enhancements in cavefishes have been shown to directly affect how they find food. The North American fishes, the amblyopsids, have a really interesting lateral line layout. From our cave tetras, we kind of see it's a bit randomly distributed across the head. Each of these ridges, hopefully you can see it, each of these ridges is hundreds of these sensory hair cells aligned. We have absolutely no idea how this translates to sensitivity or water movement versus that of the blind tetra. It does make it exciting to test their sensitivity, though. Our jumping off point for this research will start by testing the fish's ability to find food in water of varying chemistries. So we're going to start low and slow, maybe different minerals. But this is where I would love to put out there that we are always open to collaboration for water chemistry testing, water quality. We are happy to collaborate. That is definitely not my personal purview, so always looking for experts. And we are really looking forward to collecting these data. With that, I have collaborators I want to thank, funding resources, again, cavers and natural resource managers have been immensely important in our research. If you would like to contact me, that is my Alabama email, and then I have a QR code for our Extremo Evo lab website. And thank you all very much for listening to Cavefish Genetics. Thanks.